Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of KIS Organics. This is the podcast where we discuss the cutting edge of organic growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. Our guest today is Jeff Lowenfels. Jeff is the co-author of Teaming with Microbes, The Organic Gardener's Guide to the Soil Food Web, published by Timber Press in August 2006 to great acclaim and revised in 2011. It is touted as the most important gardening book published in the past 25 years and won the prestigious Garden Writers of America Gold Award for gardening books. It has been translated into Korean and French and soon Slovenian. Jeff is also the author of Teaming with Nutrients, The Organic Gardener's Guide to Optimizing Plant Nutrition, published by Timber Press in May 2013. The shorthand title is How Plants Eat and What to Feed Them. It is sure to make you appreciate the beauty of how plants operate as well as make you a better gardener and steward of the earth. His latest book, Teaming with Fungi, was just released at the end of 2016 and is all about mycorrhizal fungus. Jeff is an extremely respected and popular national garden writer. He is the former president of the Garden Writers of America, was made a GWA Fellow in 1999, and inducted into the GWA Hall of Fame, the highest honor a garden writer can achieve, in 2005. Most important, Jeff is the founder of a now national program that started as Plant a Row for Beans, the Soup Kitchen in Anchorage, and is now Plant a Row for the Hungry. The program is active in 48 states and resulted in over 1 million pounds of produce being donated to feed the hungry every year. Jeff has been a good friend now for over a decade and is a wonderful advocate for organic gardening, compost tea, and the microbes in our soil. Thanks for coming on the show, Jeff.
Sure.
I'd love to talk to you more about first off, your books, in terms of the amount of success that you've had with this. How do you recommend for users to make practical applications for a lot of the information in, teaming with microbes, teaming with nutrients, and teaming with fungi?
Sure. Well, I think each one has a different sort of indicator things that people, walk away with. For example, teaming with nutrients. I hope people are walking away with the idea that they need to test their soil. No one ever does. And then they go buy any fertilizer they think they need and put it on, and they have or microbe food either way, and they have absolutely no idea whether or not their soil or their plants need it. And so I think that's the important takeaway from that one. In addition to maybe picking up a little bit of wonder about what goes on inside those 30 trillion cells that are in every individual tree, that you're looking at, et cetera, et cetera. Teaming with microbes, I think, is something that the takeaway is that we've been doing things wrong. We've been feeding the plants instead of the microbes. We've been concerned about the fertilizer's impact on the plant instead of thinking about what everything is doing to the environment that the plant is growing in and what is the impact on the soil and the soil structure. We've been really just looking at how we grow things the wrong way. And I don't think you can read Teaming with Microbes and walk away thinking that old-fashioned post-World War II, throw on Miracle Grow kind of setup makes any sense whatsoever. So that's that one. And then the third book, Teaming with Fungi, is really, to me, sort of paving the way to the future. There is no question that when you grow plants, they need mycorrhizal fungus. and what we're learning about that is encapsulated in that book. We were just talking before the show about some of the problems with mycorrhizal fungi products, but there's no question as we move forward, we're going to solve a lot of those problems, and these are going to be items that we use, just like the average American uses Miracle Grow today, as far as I'm concerned. They'll be applied to every plant. No plants will be sold without them being added to the soil first. All nurseries will be using them, et cetera, et cetera, et cetera.
Well, Monrovia Nursery is one of the largest wholesale producers in the country. They've been using it for years now, isn't that right? Through mycorrhizal applications?
Absolutely. And more nurseries are, but yeah, Monrovia has been a real pioneer. Every single plant they sell has got a mycorrhizal partner to it. So yeah, it's the way to go. I mean, let's face it, 96% of all plants have a mycorrhizal association. Well, if that's so, then 96% of what you're growing needs to have that mycorrhizal association. And we now know that it's not necessarily there naturally. 15 years ago, we would have said, well, these things are ubiquitous. They're everywhere. You don't need to add them.
Well, if you're cloning a plant, then there really isn't a chance for it to be introduced naturally, right?
Well, it's not so much a clone situation, but even in regular normal soil, we used to think that the spores would blow all around the earth.
Okay.
After they're so small. Well, it turns out that the ectomycorrhizal, which are the ones that you might find in a big conifer forest, they may blow 3 or 4 miles. That's interesting. So if you got a nursery 3 or 4 miles away, you don't need to use ectomycorrhizal fungi. But if you're 15 miles away, maybe you do. We now know that rototilling destroys the situation. We know that using fungicides, certain fungicides, not all of them, creates a situation where you need to add them. We know that when you're planting seeds, that it makes sense to make sure that the roots are infected instantly, and so it makes sense to put the mycorrhizal fungus or fungi on the seed itself, so that you get inoculation immediately rather than have to wait. So that there are any number of, and now we know that if you use too much phosphorus or too much nitrogen, you've created a situation where the plant won't signal for the relationship to begin. And so you begin to get a whole different view than what we had 15 or 20 years ago, which is why I wrote the book, frankly. I mean, the progression of knowledge that we've had with regard to mycorrhizal fungi has been amazing, absolutely amazing. And so when I wrote Teaming with Microbes, the first edition of Teaming with Microbes, had a paragraph on them. I had to revise Teaming with Microbes in order to add a chapter on them. And then finally, I think things changed so much that it just made sense to write the whole book. So because really it's an area just like the soil food web subject matter, and really even with how plants eat, we don't learn these things in school. And we should. We learn the names of dinosaurs, but we don't learn the names of you know what I'm saying? I mean, it's sort of interesting, but it is important stuff. And if you're a gardener, I believe, and that's just sort of my opinion, I believe you have to know the science behind what you're doing.
So let's back up just a second for people who may not be as familiar with mycorrhizal fungus as you or I.
Sure.
You mentioned ectomycorrhiza, which, as you know, forms a relationship with conifers. Most of our gardeners and growers are going to be wanting Endo mycorrhizal fungus. Can you just give a quick background on some of the benefits of mycorrhizal fungus?
Sure. Well, first of all, there's 7 or 8 different kinds of mycorrhizal fungi, and they're all defined based upon how they enter the plant and how they relate inside the plant root. As you know, these fungus don't invade the cells of the plant. They are invaginated. There's an area formed around them, and then there's a transfer between the two membranes between the fungus and the plant cell. So anyway, depending how they do that, they get a different name. So there's an orchid mycorrhizal fungi, and then there's the endomycorrhizal fungi, which are invisible. They have to be stained in order to be seen. They have relatively large spores, which can be seen with a magnifying glass quite easily, and you can even sieve them out of soil. And then there's the ecto, which have very small spores. We've all seen those little mushroom spore prints. They are ectomycorrhizal fungi are visible. You can see them as they wrap around roots, and you can generally see their fruiting bodies, which are normally mushrooms or underground mushroom-like structures. Now, the endo, you're right, are generally for things that we would grow in a garden, row crops, cannabis. Those would all be ecto crops. And
endo crops.
I'm so sorry. Yes, thank you very much. Endo crops, and there are these two basic divides: the endo and the ecto. The ecto were the fungus. It was the fungus that it made plants able to grow on Earth, terrestrial land, and they were 400 million years old or so. The Endo are a little bit later. They're about 200 million years old. And, we've really discovered that even though there are many, many of them, there's about 350 mycorrhizal fungi that we're aware of now, we can only breed about 8 to 15 of them in the laboratory conditions. And fortunately for us, the endomycorrhizal fungi sleep around with different plants so that they can, the same one that will inoculate a cannabis plant will also inoculate a carrot or a potato. I hope I'm right on that one. I should really go look that up before I say that. But so we're lucky that these 8 or 9 ones that we're able to produce are able to associate with a bunch of different kinds of plants. And if you know the right one to use, then you're going to be golden. So, for example, if we may speak about cannabis, since it's legal in many states, the endomycorrhizal fungi for cannabis is readily identifiable and very available. And when you pick up a package, you can see whether or not that package contains it. It's Rhizophagus irregularis. That's one of the names. Unfortunately, because of the inability to really be able to identify what they are up until the past year or two, these things have had various different names. And now what we've been able to do, because we can now do RNA and DNA testing on them to give them their proper names, now we know exactly what to call them. And so over the next 5 or 6 years, people are going to have to change all their labels. But right now, if you buy the book, for example, you'll see certain names, and 4 or 5 years from now, those names won't be in use anymore because now we're doing DNA stuff. So, but in any case, you can look up on the web and find out what mycorrhizal fungi or fungus your individual plants might need. And then you can see if they have them on the label and put it down.
Now, when you say put it down, let's talk about best practice in terms of applying. So you'd mentioned applying it to seeds.
Right. Yeah. I like to roll my seeds in them. Well, first of all, let's back up a second. When you read the book, and of course I'm giving all the great little trade secrets away, it's the plant that sends the signal out to make the association. And the plant won't send that signal out if it's already getting what the mycorrhizal fungi will bring back to the plant. So if you're giving the plant too much phosphorus or enough phosphorus, it's not going to make that signal. If you give the plant too much nitrogen, it's not going to send out that signal. So you got to make sure, first of all, that you're not overdoing the stuff that you're already using. Once you make that determination, and again, really, you got to test your soil, see what's in it, see what kind of levels you have, etc. Everybody should be doing that. Again, that's the take home for the second book. Once you do that, then there are several different ways that you can inoculate your plants, which is sort of a funny word because you're really not inoculating your plant. The plant is calling for the fungus to come and infect it. but we use the word inoculation. So rolling the seeds is one way. And certain plants, for example, orchids, with their particular kind of mycorrhizal fungi, won't even germinate unless they're in the presence of that fungus. Not so with cannabis and carrots and tomatoes and that kind of stuff. But if you roll it on the seeds, it's right there so that as soon as you get germination, the seed root can send that signal out. You want to get early inoculation.
Yeah, so tell me what you think of this procedure. So
yeah.
The way I look at mycorrhizal fungus is as a insurance plan, essentially. So if my plant isn't getting enough phosphorus, I'll know that mycorrhizal fungus is in there as a way to make sure that happens, and fully realize that mycorrhizal fungus has a lot more benefit than just phosphorus uptake.
Right.
The way I look at it, so it's a 1 or 2 time application when I'm transplanting directly to the root zone.
Right. I roll the roots in them if I can, or sprinkle it right on the root.
Yeah, and I think when we're talking about a high value crop like a tomato or a cannabis plant, that this seems like a really great option. And also another reason to use maybe a little bit less nutrient rich initial propagation media.
Absolutely.
So that we aren't applying too much phosphorus potentially.
That's right. Yeah, that's exactly right. Your attitude is a good attitude, if that's your approach to the way you farm and garden, and that is a good approach, which is I'm taking care of the plants. I'm here. I want a little extra insurance.
Okay.
Some people's approach is, I don't really want to do anything. I want the plant to do it all itself. And that's a slightly different approach, but I think they both start the same way. You have to inoculate the plant, make sure that it's impacted or not infected, so that you can do either way.
The challenge I find is that gardeners and growers are all alike. They can't leave well enough alone. They want to tinker, and they want to feel like they're in control. It's that whole miracle grow paradigm that you talk about in your talks and in your book. So knowing that, just adding it just in case, and then giving the plants 10 to 14 days for mycorrhizal colonization before you start doing any sorts of phosphorus application as a good rule, or what?
Well, as long as you can. I mean, it can take up to 2 months for sometimes for some of these things to work. So you want to do it as early as possible, and you want to wait as long as you possibly can. But you're right. I mean, we have a tendency to think that we're in control, and that's okay. There's nothing wrong with that. But it's one of the things that you have to temper. And again, it's one of the reasons why I wrote the second book, Teeming with Nutrients, which is a book I wrote for myself, not necessarily for the general readership. And the reason why I wrote it is I really didn't know how plants ate. How did they take stuff in? How do they use the food that the mycorrhizal fungi brings back to them? What happens inside those individual cells? And I was dumbfounded by the complexity of what goes on inside cells. I was dumbfounded by the similarity of what goes on inside the cell versus what goes on in our universe. And the old Horton hears a who kind of thinking pattern is what I come away from after I read that book. And it's the reason why I'm sort of waxing about this a little bit is because as you think about your approach to gardening, as you think about what these mycorrhizal fungi are doing and when to put them on and how you want to use them and what you want them to do, your appreciation for that can only multiply once you fully understand the internal functionings of a plant. And I'm not talking about botany, I'm not talking about learning pistils and stamens and all that kind of stuff. I'm talking about, what do they do with the stuff that you give them? That when that mycorrhizal brings that phosphorus back, what does the plant do with it? And once you get that, you get an appreciation of how the plant operates. Then when you decide, gee, I'm going to give the plant everything it can, I want a little bit of insurance, or gee, I'm going to let the plant just do its own thing, you get a rationale for why you're doing what you're doing. Does that make any sense?
Yeah, no, that makes a lot of sense. I think that's great. What I find interesting about that is you and I, when we met, a decade ago now, almost. On the Compost tea side.
Well, in fact, I probably changed your diapers. No, that's not true. But I knew your parents. Yeah.
So all those years ago when we met,
I
I had no understanding of plant physiology or how a plant uptakes nutrients,
right.
And we were so focused on the biological aspects and nutrient cycling,
right?
And I'd be curious to hear, now that you've written Teeming with Nutrients, it seems like you've sort of broadened your approach to re-encompass some things like soil testing, for example.
Yeah. I mean, you and I both know that that you could most probably get anything to grow in soil if but if you change the biology. That was our approach. It was all biology. Microbiology was the base, and et cetera, and you moved on up. That's teaming with microbes, and you didn't need to test the soil. You just needed to make sure that the biology was good. But if you do test the soil, you can not only adjust the biology, you can adjust what's in the soil for the biology. It's when I got onto a listserv once that was run by Steve Solomon, who wrote a series of books that basically are Albrecht oriented, balancing the minerals in the soil. He didn't really read the book, and he gave me a lot of grief because he says, this doesn't work. You gotta feed the soil, and he kept beating up on Dr. Elaine Ingham, who he kept saying, he heard on numerous occasions, and whether she said this or not, I'm not putting words in her mouth. I'm just saying this is what he heard, that you don't need to add any fertilizer. You can grow a plant in any kind of soil based upon the biology, and he didn't believe that. Well, I still think it's true, but I think it's easier if you can also adjust the nutrients in the soil as well. You know what I'm saying? I mean, in other words, I think, yeah, I don't think you really need to add anything to. No one ever fertilizes the redwoods, as I always say. But on the other hand, if you get a redwood and you're growing it and you give it the right biology, but you also give it a little bit of, biology plant food, things do pretty darn good. So, yeah, I've changed my attitude a little bit.
You know what's interesting about that? So just to give you a little background, I actually interviewed Steve. I joined his forum a while back after reading The Intelligent Gardener.
Yeah.
Because I found it fascinating. And I too had heard Dr. Ingham's claims essentially that if you get the proper biology in place, there are sufficient minerals in all soils. Mm-hmm. So that you don't necessarily need to be adding minerals, which was what I think Steve took affront to, I guess.
Yeah.
And it's unfortunate that these two camps take such opposing philosophies. But I will tell you, in the last year, I have had Steve using aerated compost teas and experimenting with it and actually really liking the results. So
yeah.
I think.
Well, and he also asked me at one point in time to come back on the forum because he finally read the book and decided, gee, maybe this isn't so bad. So yeah, we're getting there. Man, maybe you and I just need to push a little bit more on some of these folks, but there's no reason to be so dogmatic about these things that you can't listen to somebody else.
Yeah, so coming from the background that we have, I'm learning more about soil testing, and I'm definitely a big proponent of it now. I'm finding that it's very helpful, especially when we're looking at container soils or re-amending potting soils that don't have everything that you're going to find in a traditional garden soil because it's all man-made.
Don't you find that the information is power? I mean, if you've got information, even if you don't use it, you know what's going on in your soil.
Yes.
It's power. And we know that, and gardeners know that in every other aspect. You could be a stockbroker and you're a gardener, and when you're doing your stock stuff, you get information. When you're doing the gardening, oh, my mother did this. That's not information, that's myth. And you're right. So you gotta study this stuff, and what you do is absolutely correct.
So we're not just buying the bag of tomato fertilizer because it has a tomato on it, or the cannabis bottled nutrient because it's recommended, but we're actually doing a little bit of research and have a little more knowledge so that we can make better decisions.
Yeah, you're cutting through the marketing. So, for example, I know one of your practices, same my practice. When I grow a lot of different kinds of plants, and again, I'll use cannabis as an example. I don't throw my soil away. I don't reuse my soil every single new pot. Why? I can't understand why people do that. It's because they don't understand. They don't test, and they don't understand what's going on in their soil for their plants. And the reason why they don't is because marketing doesn't want them to.
Sure.
They want you to buy new everything every time. It's crazy.
So on that note, in terms of the biological aspects of the soil, since this is an area that you're really knowledgeable in, can we talk a little bit about compost teas and nutrient cycling while I have you here? Yeah,
sure. Yeah, let's do it. Again, pretty controversial subject. Cooperative Extension Service would say we're crazy for even talking about it. Any number of people would think, gee, it doesn't work, et cetera, et cetera, et cetera. But why do you and I continue to use compost tea? Are we crazy?
Back when we started, it was an aquarium pump with an air stone. Yeah, everyone did think, Dr. Ingham was crazy and none of this was going to work. And yet now there's a ton of companies selling compost tea brewers. You see it at more and more nurseries. It's definitely catching on.
Oh, no question about it. And there's no question that the concept behind it is catching on. The biological people are coating seeds now. There are companies that do nothing but coat seeds with bacteria. Well, the origins of that are compost tea as far as I'm concerned. So, yeah, you're right. The compost tea brewers have changed. They used to use the ones that used felt and had wrong. Yeah, we've learned an awful lot about this stuff. And a lot of it's because of guys like you and Tim Wilson and of course, Dr. Elaine continues and, Carol Ann. So there's a lot of work continuing in the area. But, yeah, I love compost tea. And there's no question in my mind that it works.
Let's talk a little bit about some of the reasonings behind it. Can we give people some of the theory behind why we're making compost tea and what it does?
Sure. Well, let's start with compost. We know that compost is a very effective growing tool. Either you grow directly in compost or you amend your soil media with compost. And we know the reasons why. We know that compost contains the fertilizer bags in the soil food web, which are the bacteria and the fungus. These are the bottom of the soil food web, and when they are eaten by the fertilizer spreaders, protozoa and nematodes, which are also plentiful in compost, the resulting waste products contain nutrient, nitrogen in particular, that feed plants because it's in plant usable form. That's the first part of it. The second part we're beginning to understand is that when these microbes die, their bodies may in fact, and most probably are what create humus. Because it's one of those things that people have always been, what is humus? And it's most probably the dead bodies of these microbes. So compost tea contains, because it's compost, with energy supplied to it that strips out the bacteria and the fungus, the nematodes and the protozoa. In some instances, we also feed them at the same time and multiply them. So compost tea contains the fertilizer spreaders and the fertilizer bags that are in compost. That's about as simple as I can make it. And if we're feeding it while we're making the compost tea, we're getting those to increase in numbers and or size. And so then when we apply the compost tea to a plant, either as a foliar spray or as a drench, those microbes then have an opportunity to impact the soil food web that feeds that plant.
Perfect. That explains it well. I do want to clarify one thing that
Please.
I think is important for listeners is to realize that not all compost is created equal. So that bag of compost that at Home Depot that may have been sitting on the shelf for 6 months is probably not the same quality as something you may pull out of your very own worm bin, for example. And that makes a huge difference when we're making compost tea because
yes,
a good brewer can only replicate the beneficial microorganisms that we put into it.
That's right. And there's probably little question that what comes out of your worm bin is better than
yes.
what you buy at these other stores. And then we'll go one step further. I think a lot of us, we like the idea, although it's not from my perspective something that should be written as a law, but I like the idea of indigenous microbes to the extent possible. I like to know that the microbes that are brewing in the tea come from my area because I know that they're going to live. Now, not all of them have to be from there. So when we talk about mycorrhizal fungi, we're bringing in, et cetera, and you use Alaska humus, you bring in, a different set. But by and large, you want to use your own compost, I think. But that's just my opinion.
Yeah, no, I have heard that something like 99.6% of soil microbes are found pretty much worldwide, but I assume in different concentrations in different areas. So
right.
Things may be more successful in your soil up in Alaska than my soil here in the Seattle area. So that does make sense. And I think even if you're using, let's say you don't have your own worm castings and you know that you can source those commercially and they'll be microbially active, there's no reason you couldn't add some of your own compost or some of your own soil into the brew as a way of inoculating your soil with that.
Absolutely. I mean, I guess what we're saying is if you got to get compost from the store, make sure you get the best you possibly can. But if you can make your own, boy, that's the best way to go.
That sounds like a good rule. I like that. So from that, let's expand. So you have the high quality compost, and you're going to feed that with a food source, whether it be molasses, fish hydrolysate, whatever product you add to feed those microbes that are in the compost. We're putting it into a brewer as a way to increase the populations of those microbes and adding oxygen.
And to strip out these guys because they stick to the soil, and so you gotta strip them out, and you can't. What I always tell people is the stuff that's sticking soil together starts with bacterial slime. It's the same stuff you're brushing off your teeth in the morning. You don't get up in the morning and just, whoosh water over your teeth. You get up in the morning and you use energy to strip that slime away from your teeth. And so that's what those brewers do. Energy to strip that stuff away. Also to keep the oxygen up. Yes.
Great. So here's a question I have for you. A lot of people see the plant succession chart in your book and say, okay, well, I am growing tomatoes. Tomatoes, they're going to want a bacterial dominant soil. So they get focused on making bacterial teas. But in your book, you also, and when you talk about how the plant really controls this process of what it needs based on the exudates it's putting out into the soil. So
right.
Is there any reason not to just make a balanced tea with good nutrient cycling every time and let the plant control that?
Yeah, that makes a lot of sense. Let me back up for a second, though, because you picked the wrong example.
Okay.
It turns out, that you can keep a tomato growing for years and years and years and years. Is it really an annual? Ha ha! I don't think it is. I think tomatoes are perennials, and I mulch them with brown mulch, but that's just me.
Okay.
So, but I understand what you're saying. The plant puts out its own exudate in nature. It attracts what it needs. The plant has the ability to change its exudate to attract different things that it needs. And so, yeah, I mean, I love the idea of, first of all, when we talked about reusing soil, a lot of times I'll make my tea out of a soil or compost that I get out of a pot. It's grown a plant that I want to use the tea on. Makes sense, doesn't it? I think.
I like that. I see, looking under the microscope at our different potting soils and such, if you use good compost, your potting soil can have quite high biological activity, and there's no reason you couldn't increase that in a compost tea brewer. So I love that idea.
Yeah. Well, not only increase it in compost, but in theory, again, it's hard to tell unless you get DNA testing, etc. In theory, you're multiplying. Let's say you're growing a tomato plant. You use soil from an old tomato plant in your compost tea. In theory, you've got exudates from the tomato plant that attracted the microbes that it wanted in, and so those microbes are in that stuff you put in the microbe tea, and in theory, that's what's being multiplied, some of. So giving the plant back again the very kinds of microbes that it wanted in the first instance, which makes its job a little bit easier.
So is there any time you would brew a fungal dominant tea or a bacterial dominant tea? Because I see a lot of these recipes online on various forums and websites. I haven't seen a lot of microscope work to back a lot of these recipes.
Yeah, they're tough. And I would, if I was trying to throw something on a tree trunk that had a weep on it, I would try to make it as fungal as possible. If I'm throwing stuff down around perennials, I try to make it as fungal as possible. Whether I'm achieving the fungality, I don't really think I am, but I'm trying. And at least what I think I'm doing is I'm imparting into the tea the right kind of substrate for fungus to be able to use once it's down in the soil or on the leaf. So maybe I'm helping it in that regard. Does that make any sense?
I think so. So we always want to try fungal teas are it's hard to get good fungal growth in teas in general, but overall,
right,
that doesn't mean we're losing any bacteria. I've yet to see a tea with good fungus that didn't also have, relatively good bacterial levels.
Yeah. I mean, my thought is the bacterial levels basically stay the same, it's the fungus that you increase, so to speak, the way. That's not really true. I should take that back, but it's sort of what it feels like.
Yeah, and ultimately it's a shotgun approach. So if we put out, a good diversity and biomass of beneficial microbes, then the plant can really control what it wants. The others will die. You're not going to hurt a plant by putting a fungal tea on an annual crop like cannabis, right?
Oh, heck no. And as you say, what's not used will die and will either turn into humus or get eaten up by something else in the soil food web, so it's all good. All of those little microbes, whether they're used or not by the plant, at least the microbes themselves contain organic matter, contain nutrients. They are retained in the soil system right at the root level. And when they die, all the goodies that are in them go to somebody else. Eventually, it ends up in the plant. So it's all good.
All right. Well, while we're on the subject of soil dominance, I want to kind of throw you a curveball here.
Let me just back up one other thing.
Okay.
I think extract is also something people might want to think about. I mean, obviously I like compost teas better, but a lot of people just want to, a couple of times a year. They don't want to make a tea. They don't want to buy a Kiss brewer. They're crazy. This is the best thing in the world. But so they take some cheesecloth and they put compost in it, and they just knead it in water for 15 or 20 minutes. And they'll get good microbes in that soup. They won't get the multiple numbers that you get in a compost tea, but at least they'll get some of the stuff out of the compost, and they can make sort of an extract that way. So if they want to just sort of fool around with it a little bit, they can try that instead.
That's a great point. It doesn't have to be expensive. You don't necessarily. Tim Wilson on his Microbe Organic site has a stir method that he lists that he's tested with a microscope that people can use. So.
Right. And people really need to go to his site. It's a great site. God, he really knows his stuff. But okay, yeah.
Yeah, he's on my list for an interview as well.
Yeah.
Here's the question. When we're talking about microbial dominance in soil, and we look at that plant succession chart, and I know you're a big proponent of no-till for a lot of reasons.
Right.
I see that taking off in the cannabis community now. A lot of cannabis growers are doing that. And I just recently published a blog post on the subject. I am not completely on board, and I'll give you my reasoning, and then you tell me what you think. So we know that cannabis is an annual crop. It's going to prefer a bacterial dominant soil. That's why tilling is effective in a lot of cases. It's unfortunately why they're tilling the rainforest, is to reset that soil and destroy some of that soil structure. Now, the big problem I have with rototilling is, as you know, it creates that compaction zone at the depth of the tiller and completely destroys, fungal hyphae and all of that. But when we're talking about indoor cannabis growers in beds and containers, we're not getting out a rototiller. We're not actually tilling the soil. If we were to dig that soil to allow us to get more nutrients back into the soil rather than just top dressing continually, I've found in side-by-side tests, we seem to get better results without the same levels of microbial loss. But I would love to get your opinion on it.
Yeah, well, again, I mean, rototilling, let's back up for a second. Let me back up for a second. You're absolutely right. You want a bacterially dominated situation when you're growing cannabis, and most of our row crops for that matter. So what you're saying in theory could also apply to your carrots and your parsley, and no, not parsley because that's a biennial, but you know what I'm saying. It could apply to your row crops and things that you're an outside garden as well. And so what you're saying is, is this no-till make a lot of sense? It's not just what it does in terms of bacterial versus fungal. There are other things going on in the soil food web that we got to balance out. And we're just discussing it. I'm not saying you don't know this. So, for example, when you rototill, you chop up the mycorrhizal network and destroy it, and so it has to reestablish itself. That impacts the carbon that gets put into the soil because the endomycorrhizal fungi are coated with glomalin. That's where about 35% of your carbon comes from. It comes from those mycorrhizal fungi. And if you mess up the system by rototilling, then you've got a diminution in carbon. For example, when you cut a worm in half, as I like to say, you don't get two worms. Unless you hit it at the 18th segment, you get two halves of a dead worm. And if you hit it at the 18th segment, the first half lives, if you can believe that. So there are other things that you do to the soil food web. I understand what you're saying, and when you're talking about a container-grown plant or a small bed-grown plant, I mean, we're really not talking about rototilling.
We're taking a shovel and maybe at max double digging a raised bed is what I'm comparing.
Yeah, which is a very different thing than rototilling. You're not pulverizing the soil, are you?
No, you shouldn't be. Yeah,
you shouldn't be. Yeah. I mean, what you're doing is you're opening up the soil and you're adding in some amendments, but basically you've got big, gigantic clumps of it. So, it goes under the rule of the least disturbance possible. Least disturbance possible. Would I rototill if I was growing in big beds? No, but would I make sure that I had the ability for my roots to be able to grow apart? Yeah, I would. I mean, I'd do a little bit of disturbance. Wouldn't disturb the entire bed though. And I certainly wouldn't throw the soil away. That's the other thing.
Sure. I think we're both on board for reusing the soil. I find that it's hard to provide, because one of the other issues with indoor cannabis or cannabis grown in these beds where they're reusing the soil with no-till is that it's very hard to get in an organic system all the dry nutrients down into the rhizosphere with zero downtime. You're reusing that soil, 24 to 36 hours after you pull a crop out of it.
That's right.
And that's been the real challenge, I think, with, strictly not tilling or disturbing the soil.
Yeah, and I think people also don't understand the full rationale behind the edict of no-till. I mean, it's not necessarily because you're screwing up. Let me correct myself. It is basically because we're losing our soil because of rototilling. That's the real reason why the Department of Agriculture says no-till works and why it's so good, is because if you till in the Midwest, that soil ends up in the East. That we have dust bowl situations, and no-till we destroyed our soils by tilling during the dust bowl and we created this situation which we've lost tremendous amounts of soil. So no-till saves soil. So let's start right there. That's the most important thing about it. From a soil food web perspective, no-till prevents damage from happening to the soil food web. And the third thing is there's a general acknowledgement among organic people who grow things that it is difficult to get the solid stuff down into the soil food web. I like to tell people, gee, you put the compost on the surface. I think Elaine says the same thing. In 18 months, it's worked its way down 8, 10, 15 inches because of the soil biology.
Yes.
Well, that's fine. That's fine if you want to put the stuff down in the fall and wait till springtime. But if you're doing, as you say, a crop every 2 months, blah, 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 maybe that doesn't work so well. Then you have to balance the concept of loss of soil, which doesn't happen indoors, right?
Sure.
So you don't have to worry about that from a rototiller perspective, versus balanced off with damage to the soil food web that you want to maintain. When you're doing an indoor grow, most of the soil food web that you're really concerned about is microbiological, and microarthropod as opposed to macro. Mm-hmm. Would you agree? I think that's true. Would you agree?
Yeah, definitely.
So again, the concept of quote rototilling indoors doesn't quite do the same amount of damage as it would outdoors. You're not cutting the worm in half, so to speak. The beetles that you want to make sure that are there making air holes and you don't care about those on the indoor grow. So yeah, you can disturb the soil, but I still wouldn't pulverize it.
Great. No, it's good to get your opinion on that.
Now, let me just add one other thing, and that is when you read Teeming with Nutrients, and again, I hope everybody does. To me, it's a difficult book to read. I mean, the first and the third book, those are easy books. But Teeming with Nutrients required chemistry, and nobody likes chemistry. Nobody likes the idea of chemical formulas. And then when you start to get the idea that not only are you going to have to know chemistry, but you're going to have to know the photosynthetic formulas, then people back off. And I want to just assure everybody, I did not ever want to write about the Krebs cycle or photosynthesis, and so that you will not find that in Teeming with Nutrients. But it's a little bit thicker book to read than the other two because it does involve a little bit of chemistry. And so, but I encourage people to read it. It's really important stuff, and all three of the books fit together. And so, hold your nose and just take your time and read it. I forget why I was even mentioning that. Something important. In that book. What I say is that when you start your systems, and if you're planting in the fall, and you're preparing your soils in the fall and getting ready in the springtime, you put a band of those nutrients down just below the root system so the roots can grow into it. So you anticipate it. You put it down already, and then you put the soil on top, and then you don't rototill for 4 or 5 years because you've got a nice layer of good organics and good nutrients down there. So there are all sorts of ways to approach it. You could let's say you're doing a bed indoors, you could put your nutrients down at the bottom of the bed, and you're good for 3 or 4 grows, I would imagine.
Interesting. So that brought me to another question because it got me thinking about Teeming with Nutrients because I've read that book a couple of times, and the biggest challenge for me is the vocabulary. There's a lot of words that I'm just not used to hearing or pronouncing or even remembering what goes with what. So that's you did what you could with that, but the words are what they are. There's no way you can change the vocabulary.
And again, what a shame that we don't know those words, because they're words about what happens inside plants. You may not be a vegetarian, but that cow you're eating is. I mean, this is what runs our world, from oil, coal, everything. And we don't know the vocabulary because they didn't teach it to us in school because they didn't think it was important. It is. And for our hobby, it's very important.
I agree. I think it's a challenging book, like you mentioned, but it's well worth the time. And I mean, Teeming with Microbes is sort of the Bible of a lot of gardeners and growers nowadays. And I think it only covers one aspect, like you mentioned, of this whole process. And I think you've grown and learned and changed your philosophies even since you wrote it. I mean, that book, when did you write that book? It had to be over, 10, 15 years ago?
Yeah, 2006 is when the first edition came out. And then the revised edition came out 2000, 11, I think, and you're right. And what I discovered, and I hope everybody else discovers this as well, because I don't want to write any more books. When you write a book, it's like, ugh, God. You go in this little teeny rabbit hole, and you're down there, and that's it. You're down there until you're finished. And so, what I got out of it was enough tools between the 3 books, because I didn't know any of this stuff. I mean, I got enough tools, I think, to be able to sit down and be able to piece together things. And so when people ask a question, for example, and you're the same way, now I can see, well, let's see, from a soil food web perspective, this is part of the answer, but from a nutrient perspective, that's part of the answer. And I've got enough tools to be able to figure out what I need to know and where to go when I don't know. It's amazing. Google, folks, is the best gardening tool there is, I have to say.
Yeah, so one thing from Teaming with Nutrients that I took away was that there's a few different ways that a plant can uptake nutrients.
Right.
And the biological aspect is just one of those ways. What got me wondering a little bit more about humic and fulvic acids, because a lot of gardeners and growers are using them and swear by them. As I understand it, the humic molecule is a little bit larger and chelates or bonds with these minerals. And then the fulvic molecule allows it to be taken directly through the cellular wall into the plant. Is that correct?
Yeah, I think it.
Can you explain that a little bit better than I just did?
I think it's broken down so that it can be taken in. I don't think it goes in as fulvic acid per se, but I could be wrong in that regard. But first of all, let me suggest. I'm saying that an awful lot, aren't I? First of all, let me back up. Carbon in your soil is not coming from the humic acid, the fulvic acid. That's 13%. It's coming from the mycorrhizal fungi. It's coming from the glomalin that the mycorrhizal fungi are putting out into the soil, which is another reason why you don't necessarily want to disturb the soil too much. That glomalin is there, and when the branches of the fungi die off, of course, I guess it doesn't really matter that fungi is dead anyway, but so if you're rototilling soil that's already been harvested, it probably doesn't make that much difference. That's where your carbon's coming from. But as far as the acids are concerned, the humic and fulvic acids, they go into the plant through the membranes. And I guess to make it easy, the membrane, every cell in the plant has a membrane around it. And the membrane has different little proteins in them. And these proteins act like tunnels, or pipes, or revolving doors, depending on what kind of protein they are, to allow different kinds of nutrients and substances to get into a plant. And each individual protein will only allow one kind of molecule through it. So a phosphorus molecule will not be able to go through the tunnel that's been designed for nitrogen. And so boron, for example, has its own tunnel. Yeah, every one of the nutrients has its own individual way of tunneling into or getting into these individual. And fulvic acid goes through one of those tunnels. Probably not as fulvic acid, but probably as broken down, and then it's constituted as fulvic acid once it's inside. But I could be wrong in that regard. I'll have to look that up or think about that a little bit. But so that's why before we were talking on the show, we were talking about the idea that there are 62 different nutrients in kelp, but your plant takes in 18 different nutrients into those cells. So what happens to the rest of that stuff? Well, the rest of that stuff may be in the area that is between the cell walls. So there's the ability for nutrients to get into a root, get into the actual cell itself. So it's swimming around in the cytoplasm in the cell, and it can go from one cell in the root all the way through the plant, from cell to cell to cell to cell, not going through xylem or phloem or any of that kind of stuff, but just through the cells, and it can go all the way up to the tip of the plant, through the cells. And then there's that's called the symplast. Then there's the apoplast, which is the system of structure of the cell walls, so that if you got into a cell wall in the root of the plant, in theory, you could hike from cell wall to cell wall to cell wall to cell wall, never getting inside the cytoplasm inside the cell, never going through that membrane, and also find yourself all the way up at the tip of that plant. So a lot of the things, including fulvic acid and other things, may in fact reside in those areas of the plant where they're useful and helpful for the microbiology that's, helping operate the plant and feed the plant, et cetera, et cetera, et cetera. It's a trippy world.
So do you see a lot of benefits to, say, foliar applications using fulvic acid in a particular nutrient? I know Steve Solomon likes to say, if you think your plant's deficient in boron, then do a foliar application of boron or copper or manganese or whatever particular micronutrient we're discussing.
Yeah, I think they have their purpose. I'm not a big fan of feeding your plants foliar. And boron, I'm not sure boron even, some of the nutrients are mobile, some of them are not mobile. And they're all listed in the book, et cetera. It was a very complicated, one of the hardest things I had to do when I was researching the book was which nutrients are really mobile and which ones aren't mobile. Because some people think that you can put any nutrients on the leaves and all of them will travel to where they need to go in the plant. And that's not true. And, some people think none of them travel foliar other than, 3 or 4 or 5 cells away. So it's, so I'm not a real big proponent of it. In fact, I got some stuff sitting here. Somebody wants me to test. They sent me a bunch of stuff. It's some kind of foliar nutrient food. Okay, I'll test it. I don't know why the science would change, but I'll try it. It's made in Holland, so what the heck, Can it hurt the plant? Generally, no. And I could very well be wrong, because I usually am. So maybe it'll even help the plant. So I don't want to discourage people from trying. But I think people ought to test.
I think testing is important too. So when you talk about that, you mean setting up an experiment and having a control and making sure that you're comparing apples to apples?
Yeah, if you can, or finding somebody else who did. Again, you can go on Google Scholar and find tremendous amounts of information and tests and et cetera, et cetera. And then sometimes you can contact manufacturers and talk to them about how they do their stuff, Or you find people like you. I mean, you're testing all the time. You put your stuff up in blogs and things like that. Tim Wilson, he tests all the time, and people need to pay attention. Little less of the listserv of people talking about how big their vegetables are, and a little bit more about how they got there.
Well, I think that the cannabis industry has come a long way in that regard, especially because it was such an underground industry in a lot of states. There really wasn't a lot of research and testing and just shared knowledge beyond what was going on in some of these forums, and that information was really all speculation for the most part. So we're starting to see a lot more research and testing coming out, which I think is amazing.
Well, I think it's mandatory. It's so important. And one of the great things about the cannabis industry is that it can only go one way, and that is organic. Using that, I'll use quotes around that if you want me to. But no state regulator, wants their state cannabis to be full of pesticides and bad things. And so, and no grower, I think, wants to be imbibing their own stuff full of bad things. And so it's wonderful for the soil food web business, seems to me. And all the more reason why people need to understand what's going on, so that they can control, if that's the proper word, the environment, so that they don't need to have bad things.
And if you want to reuse your soil, it's important that you're not putting a lot of mineral salts onto it that would require a ton of leaching to remove.
Yeah. Oh, absolutely. I mean, again, I don't even think about it. I mean, I've always reused my soil. I just don't understand why people would throw away good organic soil. I guess the thought process is the nutrients have been sucked out of this soil. Really? Is that possible? A, and B, so what? It's everybody should understand. And again, this is one of the lessons out of the second book. There's so many lessons in that second book, even though, God, it's not my favorite of the three. The normal rule is called the law of return. The law of return is what operates in nature. So the reason why nobody feeds the redwoods is because when their needles drop, they go through the soil food web process. They're recycled. The nutrients that are in those leaves, are recycled and go back into the plant. Nothing is lost between the lip and the cup. When you're growing a crop, you're breaking the law of return by removing the stuff that normally would go back into the soil and put the nutrients back in again. Okay, if you understand that you're breaking the law of return, then you put yourself on parole and you put the stuff back in some form or fashion, either by using a chemical fertilizer if you're a chemical head, or by putting in a substitute organic material if you're a soil food webby. And there you go. Simple as that. It's the law of return. It's the law of return. And if you've broken it, then nothing's going to work. Doesn't matter whether you're a chemical head or whether you're, And so your soil, of course, is great stuff. It's just got that little teeny missing piece. Put it back.
Yeah, so every time we harvest the plant, we're pulling out organic matter, we're pulling out nutrients. We can replace those, and the advantages, if I understand you correctly, of replacing these nutrients and reusing the soil is that we get soil structure in our old reused soil, and we also get nutrient cycling and all these beneficial microbes working together rather than starting from scratch.
Well, I guess there's two other things I would add. I hope I can remember the second one. The first is, of course, is that you've got the microbes there in that soil already. They're dormant, perhaps, but you've got the makings of what the plant was using while it was alive. And so if you're planting the same kind of plant in that soil again, it doesn't have to try to attract, the French microbe that it needs all the way from France because it's already in the pot or in that soil bed. And then the second thing, of course, is that soil is, if you do it right, it's not only structurally mature, but it's got a setup designed or a condominium already built for new roots. So, for example, if I was growing a tomato or cannabis in a pot, when I harvest it, I cut the plant at the base. I don't pull the roots out. All of the organic matter that's in that root is there. All of the nutrients and all of that have flown towards that root system as a result of mass flow, one of the ways that nutrients get to them. All of the microbes in the rhizosphere are still there. And when I plant, the roots of the new plant will follow into the tunnels and byways and pathways of the old plant because it's easier for it. Therefore, it expends less energy. So you get a lot of benefits by reusing that soil, not the least of which is you're saving money. And, of course, on top of everything else, you're being sustainable, which is ultimately what we all need to be.
Great. So I don't want to take up too much more of your time. My last question would just be, what's probably the most common question you ask with all the talks that you're giving around the country and with all three books out now? What are growers asking you?
Can I get the book on Amazon, or do I have to buy it more expensive from you? That's the question they ask me, and of course they get over.
It's not fair, right?
No, of course they can buy it from Amazon, and should. What I don't care where you get it, you should just buy them. I think the number one question I get from growers, it's a tough one, and I think you know in a way you sort of alluded to it, and that is, it's awfully hard to be organic because you're using solids versus liquids. I don't know if that makes any sense, but there is a perception that using the chemicals, because they're in liquid form, they go right to the root system instantly, and it's easier and it's quicker and it's less muss and fuss, and it's better for your plant because they get the stuff right away. Blah, 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 blah. Versus, God, if I'm putting stuff down in my soil, it takes so long for it to get there, and unless it's bat guano, the nitrogen is not going to be available for 6 months, and all that. I think people are concerned about the timing aspect of organics, and I always tell them just calm down.
Well, so there are a lot of growers are making various types of nutrient teas now too, and certain things like fish hydrolysate, like you mentioned, bat guanos. There are other organic liquid nutrients that are a little faster acting. I would say the big difference for me is ionic versus non-ionic forms of nutrients. So
Yeah.
like you alluded to. Is that nutrient immediately available in an organic growing system? No. We may be waiting 7 to 14 days for that plant to respond.
Well, even biologically that happens. I mean, it takes 10 to 14 days for those parameciums, to start eating those bacteria down there, going crazy. So, yeah, I mean, it's there's no question there's a place for tea nutrients, no question in my mind. But if you should be starting with great composted mixed soil or pure compost with good drainage. If you start with great stuff that's got great nutrients in it to start with, because you've tested it, you've fed it, you've given it what it needs, either directly or through the compost system, then you should be good to go as long as you understand the law of return.
And I think the other thing where growers get stuck is they think that they can go with that chemical paradigm of giant plant in a tiny pot, which just doesn't work for organics. You have to give the plant enough soil.
Yeah.
If you want to grow it organically. And that's been a big challenge, I think, for a lot of growers that try and do a full cycle plant in a 1 or 2 gallon container and want to know why they're unsuccessful.
Yeah, because they're used to hydroponics, where they can. It's just different when you're using soil. And again, you have to understand how the plant operates. And you don't understand how the plant operates unless you read about it, because they didn't ever teach us this stuff in botany. And that's the downside. I never really took formal botany courses. I've looked at botany books, obviously. They just didn't teach us this stuff, how to think about this stuff. And as more and more people are growing and coming back to growing, the thing about cannabis is it's the new tomato, I think. And the plant is becoming, we were having a little blip now with Jeff Sessions, all that kind of stuff. But as the plant becomes legalized in all 50 states, which it certainly will, the idea growing these plants won't just be for medicine or for the high. They're beautiful plants. They're easy to grow. They're interesting plants. They have different colors, different leaf shapes. They're an ornamental plant, and they're bringing people back to growing things again. And you can't help but start to grow some basil, all of a sudden, because hey, what the heck, and it's a wonderful thing that's happening with this cannabis stuff. But the best thing from my perspective is I think it's altering how people are looking at botany. It's got to include the roots, as Dr. Lane would like to say, I think. It's got to include the soil food web, and now that I've written a trilogy of books and I can call myself Lord of the Roots, I so speaketh.
Wonderful. Well, I really appreciate your time today, Jeff. It was great just catching up with you, and I'm glad we got to talk about some of these things. So
well, we don't get to see each other enough, but I got to tell you, all my slides have a little kiss in it here and there. I'm always thinking of you.
Thanks, Jeff, for taking the time to chat today all the way from Anchorage, Alaska. Jeff's books Teaming with Microbes, Teaming with Nutrients, and Teaming with Fungi are all available on our website at www.kisorganics.com or on Amazon as a digital download. You are listening to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey. Stay tuned for future podcasts from leading experts around the industry. Don't forget that there's more information and articles available on our website and blog at www.kisorganics.com. And if you enjoy these podcasts, please take a moment to leave me a review on iTunes and send me your feedback and suggestions through our website contact page.